the single membrane structures of the cell structure chapter, we have endoplasmic reticulum in the first place. So I'll just start off the diagram. So this is how endoplasmic reticulum exactly looks. This is, for example, the cell membrane. This is the nuclear membrane, right? So all this region in between, this is cytoplasm. Attached to the nuclear membrane are these, right? These are rough endoplasmic reticulum, and this one non-attached, right? Non-attached ones are smooth endoplasmic reticulum, and the attached ones are rough endoplasmic reticulum. Another way to detect, and one of the most essential ways to detect how it's a rough endoplasmic reticulum, is if it has ribosomes. These white dots that we see right here, these are ribosomes, and these are very visible in those images that you see. So if you see this image right here, this is a smooth endoplasmic reticulum, and these are rough endoplasmic reticulum. What are the two ways you can detect how they're rough endoplasmic? First of all, it's attached to the nuclear membrane, and more importantly, it has ribosomes attached. So if you see these black dots, so these signify ribosomes attached. So that's why they're rough endoplasmic reticulum. Now, this is an unconventional shape, I'd rather say, a shape like this. So if you enlarge just the shape, so this enlarged structure is called this is an enlarged structure of an endoplasmic reticulum, and these yellow sac-like or rod-like structures you see are cisternae, and these red on top you see a ribosomes 80s. Initially we saw ribosome 70s in mitochondria, but these are ribosomes 80s. And this right here is an ER vesicle emerging out, and this is the final product of an ER vesicle, which further on goes to Golgi body, and then further on, which we'll study as the chapter progresses. Now, what endoplasmic reticulum does is that it is an extended system of membranous tubular network, right? Um, and it is spread throughout the cytoplasm and continued with the outer nuclear membrane. These are rough ones, and continued throughout the cytoplasm ones are only those in cytoplasm and not attached to nuclear membrane are the smooth ones. So, and they contain fluid-filled sac-like bodies termed as cisternae. So these rod-like structures I mentioned, these are fluid-filled sac-like bodies known as cisternae. And what they contain is that they contain different enzymes, and these enzymes are responsible for different modification acts, which is the sole purpose of an endoplasmic reticulum. So there's two types. There's rough endoplasmic reticulum and there's smooth endoplasmic reticulum, as I've stated multiple times. So in rough endoplasmic reticulum, the ribosomes are attached. As we saw, they're more commonly found. They're numerous in number, that means, and usually found attached to nuclear membrane. They can be not attached, but they're usually attached. Now, more commonly found, I would like to signify right here. If you see, there's one and there's two. If this is a larger one, and this is a rough endoplasmic reticulum, right? However, this one is just a small patch where there's small smooth endoplasmic reticulum. Thus, more common is the rough one. The smooth one has no ribosomes, are less common in a cell, and usually not attached to a to nuclear membrane, right? Usually not attached. Now the difference in functions. So the function of rough endoplasmic reticulum, as I stated initially in the introduction to cell structure chapter, was that it is involved in protein modification and transport in terms of ER vesicle. And the function of the smooth endoplasmic reticulum is involved in lipid synthesis. Lipid meaning fat. So that is involved in protein, and this is involved in lipid, such as steroids and cholesterols, like estrogen, progesterone, etc. So this is what concludes endoplasmic reticulum as an organelle. So, because this was a smaller one relatively, so I decided to mention another small organelle, which is vacuoles. So, vacuoles are fluid-filled storage sacs, right? So they're in two places. So they're in animal cells, and they're different in animal cells, and they're in plant cells, and they have a completely different structure in plant cells. So in an animal cell, as we've been studying from O levels, there are many small vacuoles that are present, and they're temporary bodies, such as these. There are many vacuoles, right? And this can also be termed as contractile vacuole, and we might study later on why it's called that. um the plant cell one is one centrally placed one, this right here, and it is present and it is permanent, right? So now this is the tonoplast. Tonoplast is the vacuole membrane of a plant cell. This right here, the membrane that flows, it's called the tonoplast. And what the tonoplast contains is the cell sap, is the fluid filled that we studied, that is cell sap. What it contains is water, ions, glucose, amino acids, enzymes, etc. So if we briefly see the functions, so the main function, as it is also termed as the storage sacs, it is storage, right? So it stores the sometimes the waste products, etc. um, it is involved in osmoregulation, which is the balance of water going in and coming out. It is also involved in digestion through, because it has different enzymes present inside, and involved in the support of cells. And how it does that, because of its shape right here, if we see, it is involved in the support of these plant cells. It is a part of the cytoskeleton of the plant cell, and it helps maintain its shape, which is very important for a plant cell. So these, the couple of things that I mentioned, these, which is digestion and the support, it is just for plant cells. For animal cells, it takes care of the storage and osmoregulation at times. So that concludes our endoplasmic reticulum and vacuole. I'll just reiterate it once again. Endoplasmic reticulum is an extended system of membranous tubular network that spread through the cytoplasm and continued with the nuclear membrane. They contain fluid-filled sac-like bodies termed as cisternae, and cisternae has different enzymes for different modification acts. There's two types, there's rough ER and smooth ER. Rough ER has ribosomes. They are more commonly found, and they are found attached to nuclear membrane usually. Smooth ER has no ribosomes attached. They're less common in a cell, and they're usually not attached to the nuclear membrane. The functions of both. The functions of the rough ER are protein modification and transport through ER vesicle, and the function of smooth ER is lipid synthesis, which is lipid is also known as fat. Then we moved on to vacuoles. Vacuoles are fluid-filled storage sacs. In animal cell, there are many temporary small vacuoles. In plant cell, there is one centrally placed permanent vacuole. Then we discussed the functions, which is storage and osmoregulation in plant and animal cells both, but digestion and support only take place in plant cells. So that's it for today in this video.